today's talk is kind of a brief introduction to Close-Kin Mark-Recapture. And so this talk is kind of split into three different parts. So the first part is just kind of what is Close-Kin Mark-Recapture? What's the general concept? And a little bit about that. Part two, I'm going to discuss kind of a work in progress that I've been slowly chipping away at on a simulation study for trying to do close-kin Mark-Recapture on Sable Island gray seals. And the final part of this talk is kind of a simple close-kin Mark-Recapture example on a simulated data set. So this talk, I'm kind of mainly focused on the modeling parts of close-kin Mark-Recapture, and it's kind of less on the sampling design and kind of kin finding process that hopefully will become a little more clear over the talk. So what exactly is close-kin Mark-Recapture? So I'm sure most people watching this probably have a pretty good idea of what traditional Mark-Recapture is, but just for the sake of comparison, as well as to get everybody on the same page, I have kind of this comic strip here that I've created that kind of shows a very simple example of Mark-Recapture and a very simple example of close-kin Mark-Recapture. So in kind of the simplest case of Mark-Recapture, we have a closed population. So say we have a lake of fish like here, and we want to find out the abundance of fish in the lake. So we take a sample and we mark the ones that we sampled in some way, and we release them back into the population. So in our sample, that would be the ones that we marked are the fish that are colored red. Then at a later date, we take a second independent sample and we count the number of marked individuals from the first sample that we performed. This is the recapture part of the Mark-Recapture. And then using statistics, we can use something like a Lincoln-Peterson estimator to estimate the abundance of individuals in the population. So that's just given by the sample size in our first sample, which was 5, and a sample size in our second sample, which was also 5, divided by the number of marked individuals we found in the second sample, which in this example works out to be about 12.5. And I think there was 20 that I did in this comic. So close-kin Mark-Recapture is kind of similar, but it's also kind of different. So again, suppose we have some sort of closed population at the simplest. So that means there's no births, deaths, immigration, that kind of thing. The population number is fixed. In close-kin Mark-Recapture, we would take a sample typically of adults. So that's the bigger blue fish here, as well as a sample of juveniles, which are these orange fish. So that's kind of like our mark sample. Then, kind of using the principles of genetics, we would try and find which individuals in our samples are close kin. So what do I mean by close kin? I mean like parent offspring. So mother son, mother daughter, father daughter, that sort of thing, as well as like siblings or half siblings, or potentially like grandparents, grandchildren. And so these close kin are kind of like the marks from traditional Mark-Recapture. So we have kind of 3 close kin in this example. And then similar to Mark-Recapture, we can use some sort of statistical method to estimate the abundance in the population. In this case, the adult abundance, because the way CKMR kind of works. So It's again the sample size of our number of adults, our sample size for the number of juveniles, divided by the number of close kin, or kind of the marks that we found. And then we multiply by 2 here, specifically because everybody has 2 parents. That's the idea there. So, well, in traditional mark recapture, kind of the main thing that kind of allows us to estimate things like abundance is the probability of recapturing a marked individual. In CKMR, it's the probability of 2 sampled individuals being close kin, and that's what allows us to estimate the demographic parameters like abundance, survival, and fecundity, etc. So even though the name says mark recapture, there's not really any recapturing going on. The more important bit is looking for the close kin relationships between individuals. And so these kinship probabilities can kind of vary greatly from species to species and between sexes, as well as kind of what your sampling protocol is. So like, for example, in the seals that we're going to talk a little bit about today, female seals can only have 1 pup in a year, whereas, you know, like fish can have hundreds potentially of offspring in a season, which kind of greatly changes your probability of finding kin pairs. And so that's something you really have to be careful about, and make sure that you have that right, to ensure you're getting an accurate estimate. So these kinship probabilities are kind of like the key, most important part of CKMR. And so one tool that can kind of help us think about kinship probabilities, this is what Mark Bravington, who's done a lot of work on CKMR, calls a lexigram. So on the x-axis, we just have the passage of time. The y-axis is the age of individuals. And we have kind of a line that just gives us the age at which individuals start to mature. That's this dotted age of maturity line. And so these dashed diagonal lines leading into the fish, that's kind of their lifeline, so to speak. And the vertical line coming out of it is when they are sampled. So kind of looking at this lexigram, you can see that it would be impossible for this green fish to be kind of the parent of this gray fish, right? But depending on whether or not we've done lethal or non-lethal sampling, so if we did, say, non-lethal sampling on this gray fish, and that's why its lifeline is continuing to extend on, it might be possible for this gray fish to be the parent of the red fish if it happened to survive to that date. So you kind of have to think about everything that would possibly like kind of affect the kinship probabilities that you could kind of like reasonably model. So like if, for example, in a lot of fish, the fecundity kind of changes as they grow in length. So that's something you might potentially have to deal with. You may have to deal with survival, because if you have like, they have to basically survive to be able to have offspring. And so there's just kind of a lot of things that you want to keep in mind when you're doing these kinship probabilities, and it's kind of really important to kind of nail them and get them right. So for kinship detection for genetics, I'm not going to go into too much detail about this. about how it's done, but it's kind of, you follow the rules of genetics. But typically for Close-Kin Mark-Recapture, it uses pairwise comparisons for kin detection. So basically every possible comparison that you can do between an individual to find their kin from the sampled individuals that you have that makes sense, you do. So for example, we might test against every single possible one that we sampled, and maybe we'd find one parent offspring pair within that. And so pairwise, it only considers the relationships between a pair of individuals, obviously. There's another kind of approach, which is called groupwise, which considers all relationships simultaneously. And you can kind of think about it as like trying to rebuild the pedigree or rebuild the family tree that exists. So pairwise comparisons can be less accurate, and you can kind of potentially run into issues like you could potentially identify the two fish as being the mother of one sample, which is not obviously possible. But they are a lot quicker for the large number of samples that are generally required for Close-Kin Mark-Recapture. And there's kind of ways to mitigate some of the accuracy problems, kind of both on the data end and the modeling end. So one other important thing to kind of keep in mind with kinship kind of detection and kinship relationships is that it's not possible to identify kinship relationships uniquely using only genetic data, especially in pairwise examples. So this is kind of a table giving the probability of two individuals sharing 0, 1, or 2 genes being identical by descent for some relationship status. So this column here is for sharing 0 genes, 1 gene, and 2 genes. So of course, 2 unrelated individuals are going to have 2 genes they're going to have no genes that are identical by descent, right? Like, so they have no common ancestor because they're unrelated. But you can see here from this table that for some of the relationship statuses, like half-sibling, grandparent, grandchild, niece, nephew, and uncle, they all have the same probabilities of sharing 0 and 1 genes identical by descent, right? It's all 1/2 here. And so what that means is that just using genetics alone, we can't distinguish between 2 individuals being a grandparent, grandchild, or being a half-sib, or a half-sibling, or a niece, nephew, or a niece, uncle kind of relationship, right? But sometimes you might have data on when the individuals are born or something, and that can kind of help you narrow that down. So if you know, for example, that it takes 10 years for the species that you're interested to mature, and you find a pair of individuals that are 1 year apart, have like what looks to be maybe a half-sib relationship, then you know they're not going to be grandparent, grandchild, because there's just not enough time for one of those individuals to reach the age of maturity. But sometimes you can't distinguish between them, and so there's just something you kind of have to keep in mind. So with Close-Kin Mark-Recapture, there's kind of the 3 most commonly used kinship types, I would say, right now, is parent-offspring pair, half-sibling pairs, and grandparent-grandchild pairs. And so if you're wondering why is it half-sibling pairs and not full sibling pairs, well, it kind of has to do that in most populations, where you have, well, in populations when you have random mating, you're not going to typically find a full sibling. So in like fish, for example, it's not even worth really thinking about full siblings. So they just kind of consider half siblings. So these kind of different kinship pairs kind of inform us about different things that are going on in the population. So parent and offspring pairs kind of tend to inform us about adult abundance as well as kind of fecundity or relative fecundity. And potentially, if we've done non-lethal sampling, then the parent offspring pairs can also kind of inform us about adult survival. So with a half sibling pair, the half sibling pairs that come from different birth years or between cohort pairs, they kind of inform us about adult survival and abundance. Whereas half sibling pairs from the same cohort are a little bit different, and they end up informing us about the variance of offspring from adults in that year. And so you kind of have to treat those within cohort sibling pairs differently. So you either kind of need to remove them if you can, or you kind of need to do something that will handle it being related to the variance as opposed to the other terms. And then grandparent-grandchild pairs, they all just also inform about survival and abundance. So these are just kind of some general guidelines for sampling for close-kin mark-recapture. So we kind of want independent samples. So even though it might be tempting to like take an entire nest of birds' eggs, right? Because then we would know, well, there's going to be like 5 sibling pairs in there, right? Isn't that better? Isn't more sibling pairs better? Well, if we did that, then we wouldn't really learn that much because they're all correlated. Those are correlated samples, right? So best practice is to kind of do independent sampling. You want to make sure that the individuals are mixed before you do the sampling, and that it's not like all the family members are hanging out around in the same place, because that would kind of mess up your estimates. So one thing that might be somewhat kind of counterintuitive compared to like other sampling methods is that you kind of also want to avoid kind of doing a dense sampling approach, and you kind of want to do more of a sparse sampling approach. And so this kind of is kind of a result of how you have to model CKMR models and kind of a consequence of using those pairwise comparisons. So if you end up taking like a dense sample, you often tend to like underestimate your standard errors. Whereas with a sparse sample, you can kind of better control something and get it more reasonable. So kind of like a rough guideline for sparse sampling is like something on the magnitude of like square root n of your, where n is your population size, will kind of get you closer to what you want. So typically, CKMR is performed with a population dynamics model. And so the complexity of how complex that really needs to be is going to depend again on your kinship probabilities, as well as kind of your goals for what you're interested in forecasting. And so your population dynamics model, it's going to be tracking all your numbers at age, or all your individuals in the population. It's going to be handling processes like fecundity and things that are relevant to the kinship population and survival and stuff like that. And it's just kind of controlling the population to make sure that the numbers are working with the kinship probabilities that you end up, or the kinship data that you end up providing to the model. So this kind of figure that I have here is kind of a, it's like a population at age matrix, kind of roughly. So this would be the numbers at age in the first year of the model that are age 1, and then to kind of get to the numbers at age that are age 2 in the second year of the model, some proportion of that has to survive. That's that phi here. So that's kind of all this is doing, is just kind of going through the cohorts. And so these kind of CKMR models, they tend to be fit using a pseudo likelihood approach, which is kind of related to that sparse sampling that I was mentioning, and I'll talk a little bit more about that in the next slide. So CKMR doesn't necessarily have to use a population dynamics model. So, for example, there's a paper by Rosante et al. Where they used a methods of moments estimator to perform CKMR. But there's kind of some like downsides to that approach. Like, for example, they had to have external estimates of fecundity, whereas potentially with a model-based approach, you can kind of estimate that within the model. So what exactly is this maximum likelihood and pseudo maximum likelihood that I've kind of been mentioning the last few times? So if you don't know what I've meant by maximum likelihood, so the general idea is that given some set of data, so suppose these bars is the histogram of some set of data that we observed, we're trying to find the parameters of some distribution that best matches that data. So for this example, kind of our set of parameters that would best match this data could be shown by this theta 1, right? Because it's kind of a better representation out of these three than the other two, right? So for a pseudo maximum likelihood, we don't have a true likelihood. We only have kind of an approximation to the likelihood. And sometimes this is done just because the likelihood is kind of too complex for us to figure out how to write on our own, right? Or perhaps it's just kind of too computationally time consuming. And so for CKMR, since we're kind of only doing these pairwise comparisons, and we're not really considering all the relationships between all individuals simultaneously, we're not actually getting a true likelihood. We're just getting a pseudo likelihood. So that means that it kind of doesn't kind of follow a true likelihood, which is part of why if we were to do dense sampling, that our standard errors wouldn't be quite right. But when we have sparse sampling, then we are kind of good. It's the approximation is good enough, and you'll get something that works reasonably well. So this is kind of the end of my introduction summary on what exactly is CKMR. So hopefully you have at least a little bit of a general idea on what it is and how it works. So now I'm going to talk a little bit about a more realistic example that I've been working on that's still kind of a work in progress. So this project is kind of based around the gray seals that live on Sable Island. So Sable Island is kind of a remote island that's approximately 200 kilometers off the coast of mainland Nova Scotia. And so it's home to the world's largest gray seal breeding colony. So right now, how they assess how many gray seals there are is done using a mixture of traditional mark recapture and aerial photography. So they will like actually visit the island, and they will mark the seals and count how many returned in each year that they marked. And so the goal of kind of this project that I've been working on is a little bit different than the CKMR that I was talking about in the first half, where we had both an adult and a juvenile sample. Because in this project, the goal was to assess how well a juvenile-only sampling scheme could estimate gray seal abundance. So kind of, you might be thinking, well, why only juveniles? And so the reason for this is that gray seals abandon their pups on the beach after weaning for a few weeks. So this potentially could make sample collection a lot easier and cheaper than doing the current Mark-Recapture or dealing with adults as well. So you could just kind of go around the beach and sample individuals. And so apparently there's also genetic samples that have been taken of gray seals of the past that still exist somewhere that could potentially be used for this sort of project. So it's kind of also to see how maybe if we were to use something, data similar to that, how that might help with the estimate. So again, with Close-Kin Mark-Recapture, which I've hopefully kind of driven into you at this point, it really depends on what the probability of finding close kin is. And so it depends heavily on what the life history of the species is. So kind of how long they survive and how they breed and how many offspring they have and when they mature and so on, right? So you kind of need to know all those details to get an accurate, good estimate from CKMR. So with gray seals, for example, they're relatively long-lived. They live 40 plus years. The females on average tend to live longer than the males. The females only have one pup per year, but the males can have multiple partners in a year, so they can potentially have multiple pups in a year. And there's kind of a size-based thing where individuals of a certain size tend to have more males of a certain size tend to have more partners in a year than males of another size. And so the females tend to mature around age 4, and the males tend to mature a little bit later. And so you can kind of hopefully, I think, see just from seeing this that there's going to be the kinships probabilities are going to vary greatly depending on like the sex of the parent, as well as kind of when they how they survive, right? Because the survival rate is going to be different based on the sex. So in my opinion, kind of the best way to test Close-Kin Mark-Recapture approaches is to use an individual-based simulation or individual-based model, where you kind of, in this simulation, unlike in a lot of statistical simulations, you're not just doing a model-based simulation. You're kind of actually tracking all the individuals in the population, including the births, deaths, and offspring of all of the individuals. And so because you're doing all this tracking, you know how all the individuals in your simulation are related to each other without like necessarily explicitly needing to think about, okay, well, how are, second cousins? What's the probability of two individuals being second cousins, right? It's just going to be already done for you. And it's going to be a good kind of check as well for your model. And so these individual-based simulations, they kind of basically just operate as nested for loops. So checking if individuals succeed on their role to survive or become mature, etc. So the general kind of process, which you'll see a simple example of later today as well. So we start with some sort of initial population. Then for each simulation year, we for each of the individuals that are alive that year, we check if they survive to the next year. If not, then we go to the next individual. If they did survive, then we check if they need to mature or not, if they're already mature. And then that's fine. Then we go on to have them potentially breed, and we determine, how many kids they have, if they have them, and so on. And then we go on to the next individual, and that continues for how many ever simulation years that we're interested in doing. And then we kind of get the full output of the simulation with all the individuals there. So the individual-based simulation that I developed, it was kind of modeled after the reported parameters of the current gray seal assessment model. So I tried to keep it as similar as possible to what we know about them currently, basically. And so what you see in this plot, these circles, this is what was kind of reported in the gray seal assessment model. So that's kind of like the true population abundance over time. So this year axis is kind of more going off my simulations than what happened in reality, but this is what the true kind of trend is. And so I simulated 50 populations, and you can kind of see in this figure, so this is kind of the median of these 50 populations that is simulated, as well as the 95th and 5th percentile of the population abundance from these simulations. And so because in this example, which I probably should have stated beforehand, it's all non-lethal sampling on the juveniles. And so because of that, I can kind of just resample from the same populations rather than have to do new simulations for each one. So on each of the populations that I had, I did kind of 1,000 sets of CKMR samples for them. So for each of the 50 populations, we're kind of doing it 1,000 times independently. I hope that's kind of clear. And so the way this sampling was set up is that I kind of had three kind of different sections. So the short-term sampling scheme, it's kind of meant to resemble the existing samples that I mentioned that already kind of exist, where they sampled about 500 juveniles in a year with this kind of gap period between them. That would kind of compose kind of a starting point. And then a medium where I assume that we added a few years extra of sampling starting in the near future at 700 juvenile sampling. And then kind of a long term where we just kind of kept extending it into kind of the lifespan roughly of one gray seal. And so one thing I was particularly worried about when starting this work was, well, what was the possibility of finding I wasn't expecting a huge number of say niece and uncle relationships, but I was wondering how many would be fine and if they would be kind of a serious enough nuisance that I would have to consider those kinship probabilities to deal with, even though I wasn't really interested so much in tracking them. And so this table just kind of gives the average numbers of kin pairs across the three sampling schemes, across all the samples that I took. And so it gives kind of the average number of parent-offspring pairs, half-sibling pairs, and grandparent-grandchild pairs as well as other possible relationship statuses that could show up as these other three with the same level of relatedness. And you can see they're kind of not really that much of a problem. Usually it was less than one were found in each of the simulations, each of the samples, sorry. So, and you can kind of also see another thing that's important, I think, to note is that the number of kin pairs that you find is not just linear with time, right? So you find a lot more half-sibling pairs, for example, and grandparent-grandchild pairs by increasing the number of years than just would be linear. So that's something to keep in mind. So when I kind of first tried building a model, I tried to have kind of everything. I tried to estimate both male and female abundance separately, as well as their survival. And so this model has serious issues getting the male fecundity right, and it has serious issues with the general trend. So the thick black line in these three plots, so this is from the short term, the medium term, and the long term, that's from the actual true simulated population. And then the smaller black line and the dashed lines, those are the estimates from the model fits. And so you can see that in all three cases, it tends to kind of significantly underestimate the population size. And I still kind of struggled, haven't quite figured out why that's not quite working. So I tried to make it a little more simple. And so I went for kind of like a female-only model. So with CKMR, if you have, or with genetics, if you have mitochondrial DNA, you can know, since all mitochondrial DNA is inherited from the mother, you can determine whether or not potentially a kin pair is a maternal kin pair or not. And so I tried it where I removed all but the maternal kin pairs. So I removed all the paternal kin pairs and whatnot, as well as all the potential grandparent-grandchild kin pairs. And so I had first done just a female-only model, and I was still getting kind of a biased result like you see here. I just haven't included plots of that here. But I removed all the potential grandparent-grandchild pairs. So it's not just ones that I identified as probably grandparent-grandchild pairs. I mean, I removed all the comparisons that are done that could potentially be grandparent-grandchild pairs, which for gray seals, since they mature around at age 4, would be kind of 9 years apart, right? Because you could have a gray seal that has their first pup at age 4, and then their first pup could have another offspring, their grandchild at their age 4, which would make the parent age 8, right? So, or age 9. And so this kind of ends up fixing the bias, especially in the long-term sample, but it ends up kind of throwing away kind of a lot of the data. So I think somehow the problem is related to the grandparent-grandchild that we have here, but I'm not kind of entirely sure why. So because I have this simulation, I can actually kind of check what the simulated probabilities is. And so what this plot is showing is, so the black line is kind of, that's the simulation's probability of a grandparent born in the year given by this number here in the frame being the grandparent to a grandchild born in the year of the x-axis. And so the red line is kind of my calculated probabilities. And see, they're pretty much bang on. So there might be some other issues. So I feel like my kinship probabilities are probably correct. And so I have to think a little bit more, but at least the simulation helped me identify that that part is at least okay and that I have to investigate somewhere else. So that's kind of where I am with that work. And hopefully that kind of gave you a sense that CKMR is not necessarily the simplest thing, even when you have a simulation. There's a lot of moving parts and things you have to kind of think of. But now we're going to do a simple CKMR example. So I created kind of a little simulation as well as model and data for this imaginary creature that creature. So it's an imaginary creature that I made up, and we want to estimate the female abundance. And so the main reason that it's just the female abundance is that it just makes it simpler for the example that we're doing here. And so its life history is that it kind of matures after only 1 year, and it's just guaranteed to mature after 1 year. And the average adult fecundity is believed to be constant during its lifetime, as well as the adult survival is also believed to be constant over its lifetime. So for sampling, we have kind of a 10-year program, and we're randomly sampling 40 possible mothers and 40 possible juveniles lethally. So they're not being released back in the population. They're just, yeah. And we're only interested in this example in parent-offspring pairs, and that we can identify the parent-offspring pairs exactly, so we don't have to worry about kind of dealing with our false positive rate in the model. And so the individual-based simulation that was used to generate the data for this problem is given in the file sim.r. So if you open it, you should see a file that looks like this on the left-hand side of the screen. And so I tried to keep to as much kind of base R as possible. And so kind of just good practice, as I'm sure you all know, is to set a seed to be reproducible. And then at the top of the model, the top of the simulation, I'm setting my demographic parameters. So the beta in this case is the average number of offspring that each individual is going to have in a year. And the S is the probability of them surviving from year to year. And the number of years that I'm simulating is 30. And so the first thing we do is we just create kind of an initial population, and we specify some things like where we want to keep our sampled individuals and what years we would kind of like to sample. So in this case, I'm sampling from 21 to 30. So you might be wondering why I'm simulating 30 years, but I'm only sampling from the last 10. Well, you kind of want your simulation to have a good mix of individuals first. You don't want to just necessarily sample from the initial population that you created. So by kind of doing this, I give it a bit of a burn-in. So I'm specifying how many adults and juveniles I want to sample each year, which is 40. And then I have this helper function that I created. Now what it does is it just creates new rows of our population data frame for new offspring for each year of our simulation. And so down here in this for loop it's just our simulation. So what we do is we find those that are live in the previous year that we had. We determine which of those actually survive. And if they do, we keep them alive. And if they don't, then we set them as not being alive. And then we kind of filter it out. So we're only looking at the alive ones. And since I've assumed here that they mature after 1 year, I'm just making all of the individuals mature if they weren't, and increasing the year in the population to the current year. Then to make the new offspring that they're having, I'm assuming that for now, in this population, the offspring are Poisson distributed, but they might be negative binomial or binomial. It depends kind of what's going on in your population. And that has a mean of that beta parameter that I specified earlier. And then I create the kids in that data frame. And then now I'm kind of going through and sampling out all of the adults and juveniles in the current year. So it's just randomly picking out independently 40 adults and 40 juveniles that it wants to keep, and it sets them to be no longer alive because it's lethal sampling. And then we kind of I just print out the current adult population size for simplicity, and I kind of put them back into one data frame. And so you can see after that's run, we now have a data frame called pop that, well, I'll look at the tail because that's kind of more interesting. That has kind of the ID of that individual, the ID of the mother, what year they were born, what the current year is. So it has all individuals throughout the entire life process, whether they are mature or not, and whether or not they were true. So then I just kind of get the samples from the individual base simulation, and I kind of save them somewhere in this sim.rds file, which you should have from the GitHub as well. So then we're going to need to find the kin pairs. But I think before we do that, I'll talk a little bit about what the parent-offspring pair probability is for this example, because it might make more sense why we're searching for what we're searching when we make the kin pairs. So because we used lethal sampling in this example, only individuals sampled the same year or later than a juvenile's birth year can be the mother, right? Because you can't, since they die after they're sampled, you can't have kin pairs after they're sampled, right? So I hope that's clear. So we don't have to worry about parents potentially surviving to the year that the offspring was born in this case. And so since the average fecundity is kind of the same for all adults in this example as well, we also don't have to worry about some mothers being more likely to have offspring than others, right? They all have the same average probability of being the mother, so long as they were alive in that year. And so because of that, it's then the probability that I is Jay's mother, given just Jay's birth date in this example, so that's our covariate, is just 1 over the number of adult females that were alive in that year, right? So hopefully that's not too hard to see for everyone. And you can kind of see if we did have varying fecundity between the adults, that it would, you'd have to start keeping track of that, right? So like if you assumed if they were fish that the fecundity changed with age or length or something, you would have to adjust for how many have that corresponding fecundity change, right? So now we will look at how to find the kin pairs. So that's in the file that I called make data. And so with that, you kind of can load in the simulation that we just did, which, yeah, it's fine since I already have it here, but I don't want to change my working directory right now. And so what I'm doing is I'm just adding kind of a suffix to my names of my sampled adults and juveniles so that they have an A and a J at the end of their name. And then what I'm going to do to kind of replicate that we're doing pairwise comparisons is I'm going to use this function in the reshape package, which is not installed on this computer, which is not great. But what it does is it kind of, we're going to use this expand grid.data frame. And so for every row in each data frame, we're going to end up with a kind of a, for every row in every data frame, it's going to set up kind of like a comparison, right? So it's going to expand out for every single row. So I don't think I'm explaining that super clearly, but hopefully people can understand that. So it's just kind of, we're comparing every row in the adult data frame to every single row in the juvenile data frame, right? But there are some comparisons when we just do that roughly that we know can't possibly result in valid CKMR comparisons. So we have to kind of remove those comparisons from our simulation. So for example, where the juvenile is born before the adult, right? Because then they can't be the juvenile if they were born before their parent. And we also want to remove rows where the adult was sampled before the juvenile was born because, again, the non-lethal sampling. Then, because it's just a simulation, to find whether or not the parent-offspring pairs is it's just checking if the ID of the adult is equal to the mother of the juvenile. And then, since we figured out in the parent-offspring pair probability that we only need to consider the birth year of the juvenile, that's kind of our covariate. So we can kind of cram all these comparisons that we did down into be just grouped by the birth year of the juvenile. And to do that, I'm using the dplyr command group by and summarize because that's just the easiest way that I know how to do it. So it kind of just results in this, all it is a 10-row data frame that gives you what the birth year of the juvenile was, how many parent-offspring pairs we found for that birth year of the juvenile, and how many comparisons did we have to do to find that parent-offspring pair. And so there's some more code in here where I go through doing a half-sibling comparison, but we're not going to be using it, so I think I won't cover it in the presentation. So yeah, again, this is all the data that we get that we're using for our model. It's just that much. So for the actual population dynamics for this model, it's going to be relatively simple. So we're going to have two parameters in this model. This NF1, which is just the initial number of adult females in the population in the first year of the model, and this RG, which is just the rate of growth in the number of adult females in the model. And so the parameters are going to be the things that the model is going to be moving around trying to find the maximum pseudo likelihood or likelihood 2 in this example. And so for the other years of the model, so when it's not the initial year, it just calculates the number of females in that year as the rate of growth times the number of females in the prior year. So it's really quite simple in this example. So to actually implement this model, we're going to be using RTMB. And so it's kind of the successor or spinoff of Template Model Builder, if you've heard of it, or TMB. So TMB is an RTMB. They will kind of automatically give us the derivatives of our model, which will help with finding the optimal parameters. And they can also help us calculate standard errors for different quantities. And they can also do some things like random effects for things like recruitment, which we won't be using in this example, via the Laplace approximation. So RTMB and TMB are both really great tools, and they're used a lot in fisheries and ecology for kind of fitting models like these. And so, but RTMB is kind of nicer than TMB because we can just write the model in R as opposed to C++, which is a little bit easier. So the model, that's going to be in a file called model .r. So if you look at that file, you kind of have to load in the model data, which is just this table, right? It's just the table. All we care about in this example is the pops. You want to, hopefully you have the RTMB library installed. Here, I'm just telling how many years do we have in the model. So that's just Y. And then we want to adjust kind of the model data to line up with 1 to 10, which will be what's used in the model, as opposed to 21 to 30, just to make the indexing for things easier. So all I've done here, if you look at that, I've just kind of reduced it so that now it's on, you could say, model scale instead of simulation scale. So the 21 is now a 1, and the 30 is now a 10. And so this function here that we've written called model, this is our model. It's our negative log pseudo likelihood function. And so I'll kind of briefly explain what's in there. So we specify our 2 parameters. So our initial numbers at age and our rate of growth. And the optimizers kind of prefer that they work on negative infinity to infinity scale. So because of that, we pass them in on the log scale and then convert it back using the EXP to be on the real scale, because we don't want these numbers to be negative. Then we do our population dynamics of this model. So in this case, we just have a vector that's storing the number of females in a year. And the first year of that is just going to be our parameter. So that's our initial numbers at age. Then we calculate the rest of the numbers at age for all the remaining years. So again, it's just the prior year, the number of females in the prior year times the rate of growth. And then we calculate our expected parent-offspring pair probability. And so again, since fecundity is constant among all the adults, as we showed, it's just 1 over the abundance of adults in that year, right? And then now we actually fit the CKMR observations that we used through the negative log likelihood. So we go through all 10 rows of this POPs data frame. And so we're assuming that as a part of this pseudo likelihood, that all these pop comparisons are independent binomial samples, which isn't quite true, but because we're kind of doing that sparse kind of sampling, we can kind of get away with it. And so we just give it how many pops we found, how many comparisons it took to find that number of pops, and what the probabilities is. And then here I'm just reporting some quantities from the model that I'm interested in. So that's what these report functions are. These are kind of unique to RTMB. And so it'll output my numbers at age matrix and my probabilities as well when I call a report on it. And then I can get the standard errors of some quantities like the number of adults. And so if we take this function that we run, it's just a normal R function, and we could run it giving these parameters. So you could do model params, and that'll end up getting you what the negative log likelihood is. But what we want to do is we want to give it to RTMB to create our model object so it automatically calculates the derivative for us. And of course, in this example, as I'm doing it, it doesn't want to work. It should work. I've definitely tested this beforehand, but it could be something. I didn't test it on this computer, which I probably should have done. But anyways, hopefully it'll work for you on your setup. And if it doesn't, somebody please email me and I'll fix it. And then we optimize the model. So what that does is it finds the model that best explains the set of the parameters in this data. And so the object that we should have created, but hasn't been created. I'll just try it again, hopefully. Okay, it worked this time. Great. So the model object that we've created, it has this $par value, and that's kind of our initial parameters that we're giving to the optimizer. It has the function, which gives it kind of our model function, and then it has this GR, which stands for the gradient, which is the derivative of the model. And so we give this stuff to the optimizer, which in this case is, I'm using NLMNB, and this will find the best set of parameters given the data via the negative maximum log likelihood. So you can see we got successful model convergence, and our model estimated these parameters, which are on the log scale and aren't that useful. And so you can use object.report to get those quantities from RTMB that you wanted to be reported out. And you can use the SD report on the model object to get the standard errors of your parameters and any other quantities that you might be interested in, like the numbers at age. So now we can kind of plot our results and we can kind of see how we did. So here I'm plotting the true population and the simulation. So the true population is this black line here, and our model estimate is in red, and then the dashed lines are kind of our confidence intervals. And so you can see with this one, we were pretty much bang on. Might not always be this bang on, but you can see in each year, we're roughly pretty good. So here's just kind of a prettier version of that same plot. And then here's kind of some suggested reading on CKMR if you're interested. So the first paper is arguably the most important, and it kind of explains all the concepts in much more detail than I go in, from Mark Babyn and Skog and Anderson. And then the second paper is kind of an interesting example where when you have kind of non-independent sampling due to spatial effects. And then this third paper is a recent paper that I put out with some other people that it discusses why the variance, why the half siblings are related to the variance in number of offspring, and how you can kind of use that to estimate effective population size. And the last example is just a book on population dynamics that I think is really helpful if you're interested in that.
Great. Thanks, Jonathan. Um, that was excellent. So I don't see any questions yet on the poll, um, ev.com/FE2024.
Maybe I could follow up with one question. I was really uh, sort of surprised about the recommendation for sparse sampling because it feels like um, we always want more and more samples. So maybe, Jonathan, could you talk a little bit more about that sparse versus dense sampling?
Sure, right. So it kind of goes back to um, that pseudo likelihood that you're doing, right? So you don't have a true likelihood, and you're also doing kind of these. If you're doing like a dense sample, what you can kind of end up happening is you're kind of more pulling in family clusters, like you might find like a mother, a daughter, and another son or something, right? And so you kind of no longer have kind of the independence that's kind of assumed by your pseudo likelihood, and you start kind of running into troubles there. And it can definitely give you a false sense of confidence as well.
Are there recommended cutoffs for what you would know as sparse versus dense?
There isn't really a good um, recommended cutoff that I know of. Yeah, so.
Thanks.
Well, great. Well, I guess we don't need to hang around any longer if we don't need to. But once again, Jonathan, we want to thank you for your time and for getting this all out there.